Hello, class. This discussion is intended for the two sections that were not able to have a face-to-face class with me last time. So, this will be included in the midterm exam. So, in short, class, for the midterms, you will have the chromosomes, the DNA replication, and also some of the basics of the genetic materials and also the things that I have discussed in the previous recorded discussion, plus this topic in transcription and translation. Okay? For transcription and translation, so I'll just give you an overview of the process because you will have an in-depth discussion of this one, especially on how proteins are being synthesized from this process. Okay, you will have that in your molecular biology. Okay, so remember this one. Okay, the term central dogma. So I don't know if you have remembered this term, but I think last time I have presented to you the overview of central dogma, right? So that refers to basically the central dogma in molecular level. Okay? So for the central dogma again, we have the DNA, okay, which is of course transcribed to an RNA, okay, in the process of what? Okay, in the process of transcription. And of course, once this RNA is produced, particularly the messenger RNA, okay, is the one that is playing a big role in transcription. So once the messenger RNA has obtained the genetic code coming from this DNA, because remember this DNA is in the nucleus, right? So the DNA is exclusively found in the nucleus and it cannot leave the nucleus. That's why it needs the help of the RNA. Okay, particularly again, what type of RNA? Okay, you have the messenger RNA. So the messenger RNA will get the code from the DNA inside the nucleus and will bring it to the cytoplasm. And once the genetic code is already copied from the genetic material, of course, coming from the DNA, this RNA will be converted to what? Okay, to proteins. Okay, in the process of what? Okay, in the process of translation. And of course, in determining what proteins are being produced out of the amino acids that were translated, okay, in the process, okay, you need the codon table, which I will show you later, so that you will know what are the amino acids that could possibly be, okay, produced out of the combination of the nucleotides. Okay, I don't know if you still remember the structure of the nucleotides. I have discussed that last time. Okay, so speaking of nucleotides, class, okay, I hope you still remember that one. What is the structure of the nucleotide? Okay, it has what? It has this pentose sugar, right? So you have here the pentose sugar, okay, and in particular, if it has this one, the hydroxyl group, then that is what sugar? Okay, ribose. Okay, but if it has only, okay, that hydrogen there without the hydroxyl group, or sometimes you could see just like this one, okay, so meaning to say that is deoxyribose. So I hope you watch the previous discussion that I had with you so that you will understand the structure of this pentose that I'm talking to you. So this is 1, 2, 3, 4, 5 okay, carbon atoms. Okay, so aside from the sugar, okay, contained okay, in the pentose group, this one is attached to what? This is attached to a nitrogen base. Okay, nitrogen base. Okay, remember the nitrogen base. Okay, the nitrogen base, you have, okay, this one, the cytosine, the uracil, which you can find in the DNA, and the thymine. So these are what? These are pyrimidines. Okay, or others pronounce it as pyrimidines. Okay, so this CUT here, okay, these are pyrimidines or pyrimidines. Another nucleotide, or I mean another nitrogen base, you have the A and the G, the adenine and guanine. So in this case, they are what we call as purines. Okay, just remember that purines have two syllable. Okay, so you only have two nitrogen bases, the A and the G. Okay, so that is again another attached structure to the pentose sugar. So to complete the structure of a nucleotide, this part here, is attached to a phosphate group. Okay, so in short, the nucleotide, is a collective structure, of pentose, sugar. You have the nitrogen base, the pyrimidines and purines, and of course, you have the phosphate group. Okay, so we call this as nucleotides. Okay, so that's basically what I have discussed, in the previous discussion. And I am repeating this one because, you need this, you need to memorize this, you need to understand this in order for you to understand, the structure of the DNA. Okay, another one. So we have here again, DNA to RNA that is transcribed to mRNA. This is what I have specified. And translated to proteins by what? tRNA. You might be asking me, what is the purpose of rRNA? Because again, mRNA is the one that gets the genetic code from the DNA inside the nucleus. And then the tRNA is the one that transfers the amino acids. Later, you will see how it works. So it transfers the amino acids, onto the codon found in the mRNA. How about this one, the rRNA? Where can you find this one? This rRNA class, you can find this inside, the ribosomes. And that rRNA or the ribosomal RNA is necessary, of course, for protein synthesis. Okay? So we have here the term segments of DNA. Remember this? You have differentiated, the genome from the genes. So when we are talking about the genes, it's just a segment of DNA. So they are what? They are carrying the instructions that control the production of proteins. So in short, without the DNA, your RNA cannot be translated to proteins. Okay, and genetic messages can be decoded by copying part of the nucleotide sequences from DNA into RNA. And again, the RNA now, once it has get the code, from the DNA, can now make proteins because that RNA already has the information. Okay, so this question, how does the DNA get made into RNA and that made into protein? So I'll give you an overview. Okay, still it goes back to the process, what we call as central dogma. So again, as what I have told you, the DNA is found where? Okay, let's say this is the nucleus. Okay, so the DNA is found here inside the nucleus. And remember, your DNA is double-stranded, right? So in order for it to be replicated, it needs to be unzipped. That's why in the DNA replication, you need the enzyme helicase to unzip, to unzip the double-stranded DNA. And of course, inside the DNA, you can find the nucleotide sequences. So you have here the pairing, So you have here the nucleotide sequences. So for example, if this is A, of course, because this is a DNA, this is T. Okay? If this is G, this is C. If this is C, this one, that is G. So in short, you can find the nucleotide pairs inside, the structure of the DNA. So when you get to molecular biology, we will really specify, the different structures contained in the structure, of course, or the different things contained in the structure of the DNA. The orientation of the carbon atoms, the location of the phosphate group, and even the bonds that hold, the structures together. So you will have that in molecular biology. But for now, just take note that this DNA molecule, has that pyrimidines and purines pairing. okay, in its structure. And again, okay, since the DNA is inside the nucleus, so here's the DNA, it needs, okay, the help of what again? The help of messenger RNA. So let's say, okay, the messenger RNA, so you have this color. I don't know how to change this one. Oops, there it is. So for example, you have this color of mRNA. So you have here the mRNA. So that is also having this nucleotide sequences because it will copy the sequence from the DNA. So in short, that mRNA will get to the nucleus, right? So it will copy, okay, it will copy the nucleotide sequences of the DNA. So for example, if this is G, so the DNA will be C. Okay, if this is, for example, A, then what will be the pair of that A in RNA? Because we are already talking about mRNA. The pair will already be U. Okay, how about this one? T, the pair is A, and then C, okay, the pair is G. Okay, so let's say you have that CUAG. So you have here C, okay, U, A, G. Okay, that's the nucleotide sequence that your RNA, messenger RNA, copied from the nucleus. Okay, and you also have to take note that your RNA, okay, can code for the proteins, okay, or can synthesize proteins based on three nitrogen base. So our body, of course, will read, okay, the nitrogen bases by 3, okay, and you call it as codon. Okay, so and again, let's go back. So for example, the nucleotide sequences, let me just have, for example, 6, 7, 5, 4, 5, 6. So let's say this is A, this is C, this is A also, this is G, so 3, 6, and then you have another one, you have C. Okay, so what will happen now? Okay, all of that are coming from the DNA. So in short, your messenger RNA get the information code, okay, out of the sequences of the nitrogen bases from the DNA inside the nucleus. And now the mRNA will bring this to the cytoplasm, right? And in the cytoplasm, there is a particular organelle which is again very necessary for the production of proteins. And I know that you have heard this one and you have learned this even if you are still in the elementary grade, or maybe you are in high school or senior high school. You keep on repeating this organelle. So this is what? Okay, this is, okay, the ribosome. So the ribosome, okay, is an organelle that you can find in the cytoplasm which is necessary for protein synthesis. And in short, again, as what I have told you, you have there inside of the ribosome, okay, the rRNA, which is necessary for creating this ribosomal structure. Okay, what will happen now? So now the mRNA has that sequence. So that sequence will be brought, okay, will be brought to the ribosome. So the mRNA will bring that structure to the ribosome. So now let's say you have here the structure. Okay, let me just select a darker yellow. Okay, so you have there, okay, the mRNA, and then still you have that sequence. Okay, so for example, you still have the CUA, then GAC, AGC. Okay, so again, as what I have mentioned, okay, your mRNA will, okay, read the nucleotides, okay, by 3s. Okay, so now, okay, the mRNA is already in the ribosome. But take note, class, there is what we call as start codon and another one stop codon. Okay, so the start codon, okay, we have the start and stop codon. So the usual, okay, start codon, and you will recognize it because, okay, it is in a UG sequence. So if you have here the mRNA, okay, it will start transcribing proteins, okay, when it has recognized the AUG. So in short, you have here the A, the U, and the G. And the very first protein that will be, the very first amino acid that will be transferred, okay, and that will be brought to that start codon is the protein methionine, okay, or amino acid methionine. Okay, I am just very specific on the usage of the term because again, methionine is an amino acid necessary for creating proteins. So that's why I'm explaining that the very first amino acid that will be brought to the mRNA to pair up with the start codon is a methionine. So how can that protein, okay, synthesis happen? So you have there the yellow structure, the mRNA, okay, it starts copying the nucleotide sequences, okay, with the start codon, the AUG. And now, okay, there is a structure, okay, let me just select again another color. So let's say we have the tRNA, okay, the tRNA. So let's say this is the tRNA. So this tRNA here, okay, you have the tRNA. It also has these three structures which is corresponding to the sequence, okay, of the nitrogen base that you can find in the mRNA. And that tRNA, this is the tRNA. This tRNA class will again carry what? Will carry the amino acid. So let's say you have here the amino acid. Okay, that's the amino acid. Okay, amino acid. So again, the very first amino acid that will be carried by the tRNA to the mRNA in the ribosome is based on the sequence of the start codon. So now, for example, you have here the tRNA. So the first one that will be carried by the tRNA, so you have here the tRNA. So the first one that it will carry, okay, this one. Oops. Okay, the amino acid that it will carry, the very first one again, as what I have said, methionine. Okay, remember the sequence of the pyrimidines here, the purines and pyrimidines here. Okay, AUG. So the tRNA now will be copying the nucleotide sequences from the mRNA, and it will bring, okay, three nitrogen bases. Okay, so here the AUG, CUA, GAC, AGC. So I just made up that one. Okay, except for the AUG because the AUG is really the start codon. So as what you can see, I am illustrating it by 3. So for mRNA, okay, we call it as codons. Okay, codons. Okay, so by 3, okay, that you can find in the mRNA. But for the tRNA, how do we call it? Okay, we call it as anti-codon. So now, the tRNA will copy the sequence coming from the start codon because without a start codon, then we cannot, okay, start the process of translation. So now the tRNA will find the pair of A. So what is the pair of A? Okay, this is already U because we are already talking about RNA. The pair of U will be A, and the pair of G will be C. So UAC. You remember that one because we need to check that later in the codon table. So UAC. And now, once this tRNA already brought this amino acid to the ribosome, so this one, this is methionine. Okay, now the tRNA will get another nucleotide. Okay, and what will happen? Okay, it will start adding again. So you have here the tRNA, and then it will bring another amino acid. Let's say this one. It depends on the sequence of the nitrogen bases in the mRNA. So now the tRNA will follow the sequence until, okay, it has brought a lot of amino acids. So you have, for example, G, you have A, and then you have U. Another one in this case, okay, the tRNA will pick up another amino acid. Okay? So, for example, you have here, okay, the tRNA and then another amino acid. Okay? And then the tRNA will again follow the sequence. So, you have the C, the U, and the G. Right? Another one, okay, take note that as your tRNA brings the amino acids, okay, to the ribosomes, there is a bond that holds the amino acids together. And you know this, you learn this in biochemistry. What bond, okay, holds the amino acids together as your tRNA transfers the amino acids to the ribosome? So that is, of course, a peptide bond. So in short class, your tRNA, okay, will start adding the amino acid until such time what? Remember, you have the start codon, the AUG. It needs a stop codon. So let's say you have a very long chain of amino acid. Okay, you have this one by 3, and then by 3, and then by 3. So you have, for example, A, and then U, A, G, and then this one, U, G, A. So the tRNA class will continue adding that amino acids, that nitrogen bases, that pyrimidines or purines, okay, depending on the what do you call this? The sequence of the amino acids. Okay, the nitrogen base. Okay, I'm sorry because I'm getting lost with these colors. Okay, so you have here, for example, AA. This is, let's say, G. So now you have there, okay, this one, C. So in short, the tRNA will continue that one. So finding the pair, okay, until such time it finds the stop codon. So this stop codon here, such as this UGA, the UGA, okay, that is a stop codon. Actually, there are 3 of them, okay, stop codon. So when the tRNA already recognized this one, this is already a stop codon. So that is the time that the tRNA will stop adding nucleotides. So the sequence will be only until there. So you have A, C, and then U. And then, of course, the amino acids, okay, will be linked together, okay, by a peptide bond. So you have their different amino acids, okay, and then linked together by peptide bond. So the length of the amino acids class will depend, will depend on the sequences of the nitrogen bases that your tRNA follows, okay, out of the sequences that you have in the mRNA. You get my point? So that's how proteins are made. So these are amino acids linked together by peptide bond. And of course, we call this as polypeptide. Okay? These are proteins. Okay? Necessary. Okay? Of course, for a lot of function in our body. So that is how transcription and translation happens, class. And I hope you really understand the process. Okay? So that is how the proteins are being made. Okay, another one. So I have also presented to you the differences of the DNA and RNA last time. So this is just a recap. So the DNA contains deoxyribose, this one ribose, but both of them, they are pentoses. Okay, again, the ACGT, okay, the T is replaced by uracil in the RNA. Double strand, that's DNA. Single strand, that's RNA. Stays in the nucleus as what I have told you. That's why it needs mRNA, because it's the mRNA that will send, okay, the code to the cytoplasm. Okay, another one. What are the 3 main differences between DNA and RNA? So, of course, again, ribose and deoxyribose. So, the sugar. Next one, the strand. So, DNA is double-stranded. RNA is single-stranded. Okay, and then the thymine and uracil replacement. In the uracil is being replaced. The uracil replaces thymine in RNA. Okay, another one. So again, there are 3 types of RNA. Okay, 3 types of RNA. So you have mRNA, okay, mRNA, you have tRNA, and you have also rRNA, the ribosomal RNA. Okay? So, for the messenger RNA again, what does it do? So, it is made from where? The mRNA. Do not be confused with this one. Made from DNA in the nucleus. You might be confused. Where is the mRNA coming from? Of course, that's from the nucleus. It just travels out of the nucleus to the cytoplasm and of course find the ribosome where it can place the nucleotide sequences there. For the tRNA, as what you can see in my illustration, it transfers amino acids which are connected by peptide bonds to the ribosomes. Okay? And then the rRNA, as what I have said, is just part of the ribosomes where proteins are made. Okay? So those are basically the 3 different types of RNA. Another one, this is just showing you the structure of the mRNA. And then, of course, the rRNA, okay, made up of proteins, ang ribosomes, and then, of course, it has also the rRNA. Okay, the tRNA again, what is this one? The amino acids, okay, carried by the tRNA. And take note, how do you call this one? This is the anti-codon, the one that you can find in the mRNA, that is the codon. But in order for your tRNA to bring the ATGC or AUGC to the mRNA, it needs a start codon. And in order for it to stop copying and bringing amino acid, okay, it needs a stop codon. Simple as that. Okay. Another one, this is just the concept map. Okay, it's very easy because now you understand the entire process. So the RNA can be messenger RNA, okay, also called mRNA, which functions to carry instructions from DNA to the ribosomes, while the rRNA is also called the rRNA, which functions to combine proteins to make up the ribosome. So it's important for the structure of the ribosome. The transfer RNA, also called the tRNA, okay, functions to bring amino acids to the ribosomes. Okay? And when we talk about transcriptions, it's just simply DNA to what? To RNA. Particularly what RNA? Again, mRNA. So RNA molecules are produced by copying part of a nucleotide sequence of DNA into a complementary sequence in RNA. This process is called transcription. Transcription requires the enzyme RNA polymerase. Remember, okay, in DNA replication, the moment the DNA is unwound, remember, it needs a primer class, right? It needs a primer. And it's the DNA polymerase that will add nucleotides, okay? And this is now creating the daughter strand, the new strand. So that is for DNA replication. That's DNA polymerase 3. Because I have emphasized to you that DNA polymerase 1 is necessary for proofreading. Okay, but in adding the nucleotides to the unzipped strand of DNA, if you have watched my previous discussion, then it needs DNA polymerase. But for transcription process, because we are dealing with RNA, particularly the messenger RNA, it needs RNA polymerase that functions the same way the DNA polymerase adds, okay, the nucleotide sequences, okay, in the growing strand of the mRNA. Okay? Another thing, so how RNA is made from DNA. Okay, so in eukaryotes class, you can usually see this Tata box. Okay, that's how they call it. Okay, so just take note of that one because that is necessary. And that is where the RNA polymerase is starting to add the nucleotides, okay, to copy the code from the DNA. Okay, so this one. So this is the summary of the steps. So the RNA polymerase binds to the promoter site. So this is what I'm telling you, the Tata box. So that is where the RNA polymerase will bind to, okay, on the DNA. So that Tata box, you can find that in the DNA. So this is still color green. So that's part of the DNA, which again, the RNA polymerase will bind to Remember, in the DNA replication, you have the RNAs, okay, which will make the RNA primer, right? Or RNA primase, which will make the primer rather. Okay, so in short, in transcription, you have in particular, in eukaryotic cells, you have the Tata box, okay, which the RNA will bind to. And then, of course, it's the RNA polymerase that will add the RNA nucleotides complementary to what? Okay, DNA strand. So in short, it's the RNA that will copy, okay, the DNA nucleotide sequences. And maybe you can already connect why is it that RNA is just single-stranded. Because again, it's just, okay, made for copying the code coming from the DNA. Okay? And then, in particular, the mRNA building is complete when the RNA polymerase reaches a termination or the stop site on the DNA. And now, the mRNA, okay, will leave the nucleus and will go to the cytoplasm. However, class, you need to take note that before the mRNA could leave the nucleus, it needs to be what? It needs to be edited. Okay? It needs to be edited again before it leaves the nucleus and before it carries, okay, or before it can carry the nucleotide sequences to the ribosomes. And take note, this is what I have emphasized to you. DNA never leaves the nucleus. Okay, that's why it needs RNA. Okay, another one, this is just showing you the RNA polymerase. Okay, it is the one adding the nucleotides to the growing mRNA strand. Okay, and take note of this one, uracil is for RNA only. For you to remember that one, out of the pyrimidines and purines here, it's only, okay, as what you can see in the spelling, it's only the uracil that contains letter R. So it's an indication, and it's like a mnemonic for you maybe, okay, that you can remember that it is the one, okay, that you can find only in the RNA. Okay, another one, so AT, again this one, AU or CG or GC, etc. Okay, TA. Another one, okay, so you can transcribe this. So the T pair is U, so this is the DNA, and this is already the mRNA, right? So that's why you already have the uracil here. Okay, simple as that. And then, is all the DNA transcribed into mRNA? No, again, because I have said, before the mRNA can leave the nucleus, it needs to be what? It needs to be edited. So only certain sections of the DNA are made or transcribed into message RNA or messenger RNA, and only part of the mRNA is actually used and sent out of nucleus to meet up with the ribosome, and this is what you call as the editing. So how does that editing happen? So you have the term exons and introns. So these are areas that you can find in the mRNA strand. So take note of that one. So introns are cut out. Okay, introns. They are removed, okay, before leaving the nucleus. So what is left is only the exon, okay, or the exons. Okay, so just like this one. Okay, so you have the intron. So again, they are cut out, they are removed. That's why, okay, you no longer have that one. But in pre-mRNA, of course, there is, okay, the production of this one. Okay, there's the removal of that intron. That's why you have this entire mRNA strands containing only exons. Okay, the exons are the spliced together to form mRNA. And take note, the introns are cut out of RNA molecules. Okay, so this one, introns, they will be removed. Okay, that's why you have these exons. Okay, and that exons will be the one, okay, to be translated to proteins. Okay, so RNA to proteins, we call it as translation. Okay, and take note, it's the mRNA now in the ribosome that will code for certain amino acids carried by what? Carried by tRNA. Okay, and the strings of amino acids, we call it as proteins. And again, the amino acids are held together by peptide bonds. Okay, another one. Translation. Translation is the decoding of an mRNA into a polypeptide chain protein. mRNA to protein. So what transfers it? Of course, transfer RNA, and it takes place on the ribosomes, and the process of translation is really to produce protein. So this is the amino acid, this is the tRNA. And how do we call this again? There it is, the anticodon. So translation steps, you know this already. Messenger RNA is transcribed in the nucleus. So transcribed meaning to say that's DNA to mRNA in particular. And it enters the cytoplasm, but before that, it needs to be edited, right? The introns are removed. Repetition class will create memory. That's why I keep on repeating this one. The introns are removed. And then, and then that introns will, of course, what is happening here? That introns now, will make up the mRNA strand, where it is the one that is attaching to the ribosome, to begin the translation process. So this is still the nucleus where the DNA resides, and you have here the mRNA being transcribed or decoded, from the DNA genetic code. So initiation, you know that already, and then there will be elongation as the tRNA starts to add nucleotide sequences, and bring amino acids to the ribosomes. And again, as what I have told you, we have the start and stop codons for the tRNA, to be signaled, and to know where to add and where to stop adding the nucleotides. So this one, it's still the same. So the mRNA attaches to a ribosome, and then the tRNA will bring this amino acid to the ribosome. And of course, now I will be emphasizing again this one, the AUG, that is the start codon. And I have told you to remember this one, the UAC. UAC, because the first amino acid, brought to the ribosome, because of this start codon, is the amino acid methionine. And of course, again, the tRNA will continue adding, anticodons. That's why by 3, until such time it finds the stop codon. So we have here each tRNA molecule carries only one kind of amino acids as determined by what? Anticodon. In addition to an amino acid, each tRNA molecule has 3 unpaired bases. And these bases, we call it anticodon, are complementary to the mRNA codon. And to determine, again, what amino acid is coded for, what will you look at? The codon chart. I have mentioned this at the very first part of this discussion. So this is just, again, the summary of the translation process. So mRNA leaves the nucleus and enters the ribosome. They are edited. mRNA codons read, and tRNA brings the matching what? Amino acid to the ribosome. And then the tRNA anticodon is complementary to the mRNA codon. Amino acids are strung together like beads on a necklace, and they are held together by peptide bonds. That's why you have 1,000 or more amino acids, produced in that process, and we call that as proteins. So this one, genetic code. So the genetic code is the language. So that's the layman's term there. The language of mRNA instruction, and the code is written using 4 letters. The purines, and the pyrimidines. So you have AUCG. And now, this is the codon table. You convert, the codons, to amino acids. Like for example, the AUG, the A, the U, and the G, you have the methionine or methylamine, the start codon. So when you use this codon table, so let's say you have the A, the U, the second letter, and the G. So the A, U, and then the G. So this is letter G. So AUG. So that's, okay, the methionine, okay, or the start codon. So we only have one start codon, and that is the AUG. That's why I told you earlier that the tRNA always add first the methionine because, again, of the sequence of the start codon. Okay, for the stop codon, you have the UGA. Okay, I have told you we have three: the UGA, the UAA, the UGA, and UAG. Okay, the UGA, UAA, and UAG. So those are the stop codon. So how will you read this one? For example, in our illustration earlier, okay, I have here, oops, it's too far away already. Okay, so let's say, okay, in our very first illustration, okay, the second is GAU, the third is CUG, GAU and CUG. Okay, so we have the G, AU, and CUG. Let me just go back to that one. This one, GAU, that's the second, and CUG. So what's the amino acid of GAU? Okay, the GAU, okay, let me just erase this. GAU, so we have here the G, okay, and then the second position, we have the A, so GA, and then of course, you have to align that one, A, and then G, you find letter U, so it's here, GAU, so GAU, so that's, again, aspartame, okay, so GAU. How about the CUG? CUG, okay, the C here, the U, and here's the G, CUG. So that's leucine. So I think this is easier. Okay, the first one is GAU. So G, and then A, and then U. So that's the aspartame, aspartic acid. How about the CUG? You have the C, okay, what's the second letter? U, and of course G. So that's leucine. So you can make use of this one. This codon table or this circle, okay, codon table, codon ring, okay. So this is just another codon table, okay, emphasizing, okay, the UGA, the UAA, and UAG stop codon, and you have the start codon. Okay, just a summary of the protein synthesis class, okay, out of the transcription and translation process. Okay, so here's the mRNA, here's the ribosome. I hope you recognize the structure. Okay, so that's why, okay, if you read other books, you can say that the DNA is the master plan. Okay, because it's where, okay, the RNA will get the original code, okay, from the DNA to prepare the RNA. And the RNA are the blueprints because it's the mRNA that will be followed by the tRNA. Okay, and that all, okay, creates, okay, of course, proteins. So you have, for example, you transcribe this one, okay, so this is how it is made. So this one, the white letters, you have the TA, so that's coming from the DNA, the original strand. This is from the nucleus, and then of course the RNA, the mRNA will copy that one. So you have the ATUAGC, and then etc., etc. And now the tRNA will bring, okay, the sequences of nucleotides depending on the sequence of the nucleotides in the mRNA. Remember this one, the UAC? Okay, so now, what's the RNA there from here? UAC earlier, we see in the table, the AUG earlier, that's methionine. And then, how about this one? The UCA. Okay, UCA, where is the table? U, C, and A. Okay, serine, this one. Okay, this one, serine. Okay, UGG, okay, you follow the mRNA sequence because that's where the tRNA will bring the amino acid. So the UGG, okay, you look at the table, okay, the U, G, and G here. So UGG, that's tryptophan. Okay, hope you memorize that from your biochem. Okay, etc., etc. And remember, the UAC, because you have the UGA, the UAA, and of course, the UAG. Okay, that's for the stop codon. Okay, that's why it's stop here. So, I think that ends my discussion, class, and I hope that you really understand the process of protein synthesis out of the central dogma. So, whenever you are reading your exam questions, I hope that you will not only memorize, okay, the things that I have discussed to you, but as well as try to at least understand and analyze the given situation, okay? And study the codon table, class, because part of your exam contains that codon table which you will analyze. Okay, thank you, class. God bless.